And while we're waiting for people to join us, just like to add a couple of reminders that our next roundtable, scientific roundtable, will be January 6th of 2025. Hard to believe this year is gone already. Dr. Paul DeCane from Northwestern will be presenting. I want to take this time also to, hope everybody had a wonderful Thanksgiving holiday, and we'll have a wonderful holiday coming up in December. Take a bit of a break on some of the stuff during the holiday season, but we're back at it at January 6th again. There's several new cell models that have become available for studying PKD up on our website. Encourage you to go up there, take a look what we have available. And with that, I'm going to introduce today's speaker, Dr. Mahjoub from Washington University in St. Louis. He is an associate professor there, and he's going to tell us today about defining pathomechanisms of nephronophthisis-related renal ciliopathy. So Mo, turn it over to you.
Thank you, Brad. I hope everyone can hear me okay.
And he's good.
Okay, let me see if I can share my desktop. Can you see this PowerPoint?
Looks perfect, yes.
All right, great. Okay. All right, so, and you can see my arrow there?
Yes, all good.
Right. Perfect. Yeah, so thanks for the invitation, you guys. I really appreciate it. This is a wonderful community, and it's always nice to be able to kind of present our work and get some feedback from you all. And so I'm going to tell you a little bit about what my lab's been working on. I'll leave you with some following up on. And there's really two things that I'm going to talk about for today's talk. First is this focusing on kind of characterizing pathways that are disrupted in early kidney development in the early onset renal ciliopathy. And we'll talk more about that. I know that this group is usually more focused on PKD, ADPKD, and ARPKD, but if you all indulge me a little bit, we're going to be talking about a little more of the rare renal ciliopathies. So I'll tell you a story about that and our new mouse model of this. And then I thought, since this is the PKD RRC, and as a resource, I'll highlight our recent paper on using expansion microscopy on preclinical and clinical renal samples to sort of give you a hint of what you can do with this approach. And hopefully you all will try it out. So, my lab works on cilia and cilia-associated diseases called ciliopathies. You've heard from multiple people over the years of this meeting, I'm sure, about what these things are, the different organ systems that are affected. For today's talk, of course, we're going to be focusing on the kidney-associated phenotypes of this disease syndrome. And of course, it all has to do with our favorite organelle, the primary cilium, which has been, a heavy focus of the kidney field for many, many years because of its involvement in kidney development and, of course, in the pathogenesis of PKD and other cystic and fibrotic diseases. There's been a lot of work in identifying the signaling pathways that function through this thing, the diseases that are associated with mutations in proteins that are part of the cilium. I want to focus you today on this structure at the bottom, the basal bodies or the centrosome, the structure at the base that builds this thing. It doesn't get as much love by the, I would say, the cystic disease or the PKD community in large. Most of the focus has been on cilia, and for good reason. But the centrosome sort of gets a second role usually in this, and it's been not as well studied with regards to kidney development and kidney function. So we thought, we've been very interested in centrosome biogenesis, so we thought we'd focus on that for some of our studies, and that's what I'm going to talk about today. So just a quick overview. We know there's, over at least 1,000 different proteins that make up these complexes. There are at least 200 genes and counting that have been discovered to be causal in ciliopathy patients, and over 60 of these genes mutations have a kidney phenotype associated with them. Interestingly, out of all of the mutated genes, about 20% actually encode centrosomal proteins and not cilia proper proteins. And many of these mutated centriolar centrosomal genes are important for maintaining centriolar centrosome structure, or they're involved in centrosome biogenesis, or in regulating the functions of the centrosome. And several of these mutated centrosomal genes are found in patients with these rare cystic and fibrotic kidney diseases, like nephronophthisis-related ciliopathies, which is the focus of today. So as I mentioned earlier, there's been a lot of focus on the role of cilia and ciliary signaling and cilia function in kidney development and disease, but much less about centrosomal defects and how they disrupt kidney development and how they drive disease phenotype. So that's where we kind of have been focusing on in my lab. And I don't need to introduce this slide too much to this audience. You all know the differences between the very prevalent cystic disease, the ADPKD, compared to these rare ones. There are, morphological, clinical, molecular, cellular differences between the two diseases. The focus for today, of course, is going to be on these NPHP-RC. These are part of syndromes like Joubert syndrome, Jeune syndrome, Bardet-Biedl syndrome, Meckel syndrome, and of course, nephronophthitis itself. They're individually rare. Incidences are 1 in 20 or 100,000, 200,000. But when you actually add up the incidences across all of the different ciliopathies that have this kidney phenomenon, the incidence actually goes up to about 1 in 2,000. So it's pretty common. Unlike ADPKD, which we all know is very slow and progressive, these diseases are very early onset. So clinically, they're much more severe. They reach renal failure very early within the first or second decade of life. These come with congenital kidney developmental defects, whereas ADPKD kidneys typically develop normally. They're smaller kidneys, of course, whereas ADPKD kidneys get very, very big and very cystic. And these diseases are predominantly fibrotic with cysts as secondary. So this disease, of course, has been very well studied or heavily studied, and the link to cilia established. This hasn't been studied as well, and the link with regards to centrosome biology, not established at all, I would say. There's only like a very few papers that have looked at centrosomal genes in the context of this at all. So our main objective was to kind of define the cellular and molecular defects upon mutations that disrupt centrosome function during kidney development and compare it to loss of ciliary signaling function or loss of cilia structure itself. So just to kind of try and tease apart the additional contributions or roles of the centrosome in the disease outcomes. So for that, we needed a gene to use as a molecular handle to study. And the one we went with is this protein called CEP120 or centrosomal protein 120. It's 120 kilodaltons. We picked it for several reasons. First of all, we have characterized this thing for a long time. So I first started working on this protein when I was a postdoc in Tim Stern's lab, defined its role in centrosome duplication during the cell cycle. And so we know quite a bit about its function at the centrosome. We've defined what it does. We know where it is. It's enriched on daughter centrioles. Its function is to help when centrioles duplicate in S phase and have to form new centrioles to duplicate the existing centrosome. CEP120 is heavily involved in that duplication. And so if you have a mutation in CEP120, you fail to duplicate your centrioles in S phase. And so in mitosis, you don't have a complete centrosome, and daughter cells will only inherit one centriole and not two. And that's called centriole loss or centrosome loss. And in the next cell division, again, because you're missing CEP120 and you can't duplicate your centriole, that one centriole cannot duplicate. And so daughter cells will inherit either 1 or a 0. This phenomenon is called centrosome loss. It's a common phenomenon in cancer and in other ciliopathies like microcephaly and other organ systems. So we've studied the process pretty well. And in the process of studying CEP120, we've developed a lot of reagents. So we have a lot of tools against it, and including a mouse model. So it gives us a handle to study this really well. Importantly, this thing is mutated in cohorts of patients with the disease syndrome that we care about, with this nephronophthisis-related renal ciliopathies. And indeed, these patients develop these congenital renal dysplasia, typical of nephronophthisis. Their kidneys are small, they're mostly fibrotic and less cystic than ADPKD, and they reached ESRD within the first 10 years of life. So CEP120 gives us a physiologically relevant protein to study these early onset severe nephronophthisis-related ciliopathies, and also gives us a role to study a way to study the role of the centrosome in this whole process. So what we decided to do, we made a fluxed mouse, and what we decided to do was to try and tease apart genetically where a mutation in a centrosome gene might have the most impact on the disease and on the congenital developmental defects and also the disease progression. So we decided to use early developmental Cres and separately knock out CEP120 in the three main progenitor population of a developing kidney. So we decided to knock it out in the metanephric mesenchyme, in the ureteric bud epithelia, and in the stromal mesenchyme. And the idea was to look at what happens to nephron development and what happens to the development of the stromal populations, but do it surgically instead of ablate it in all of the cell types and sort of see the overall outcome, which we can do, of course, but we wanted to take a sort of a surgical approach and asking which progenitor population gives us which sort of outcome when we knock out this gene. So two postdocs took on this project. The first, Tao Cheng, knocked out CEP120 in the mesenchyme and the ureteric bud epithelia. So he was looking at nephrons and nephrons in the stromal progenitors. These two stories were published earlier this year. So you can look, if you're interested in all of the details, you can look at them in greater detail. I'm just going to highlight some of the main take-home messages of the two stories. So we used the 6-2 Cre and HOXB Cre to knock out mesenchyme or the UB progenitors, respectively. These are both the developmental Cre models, so these are not inducible. So they will get knocked out developmentally when these Cres are normally expressed based on the promoter. And so we crossed them separately to the CEP120 flox mouse. And just as a comparison, we crossed them also to to PKD1 flox to knock out, to disrupt ciliary signaling in the same two progenitor populations, just as a comparison. And what I'm not showing you on here is we also included the IFT88 flox in these crosses so that we can structurally disrupt cilia, but keep the centrosome intact. So now we have three systems where we have, cilia and centrosomes intact, just missing the polycystin, or cilia missing, but centrosome intact, or centrosome and cilia are missing. So we can do a comparison. And I'm going to show you the data for CEP120 and PKD1. The IFT88 looks very much like PKD1, so we didn't include it in a lot of the analysis. So Tao first went ahead and ensured that, in the mesenchyme or in the UB, he could see significant reduction in the percentage of cells that express CEP120 in the respective populations. And when he does that, we should see loss of centrosomes as the cells divide, and that's exactly what he sees when he quantifies centrosome number in the two populations. So he's able to knock out CEP120 in centrosomes, and of course, this should not happen in the PKD1 knockout mice, and it doesn't. So both CEP120 and centrosomes are both present in the respective progenitor populations in the PKD as expected. So what happens phenotypically? Well, here's the couple of embryonic developmental time points up to birth for a wild-type kidney. Here's the 6-2 and the HOXB kidneys. You'll notice that as early as E13.5, we can already see these kidneys look a little bit weird. They look dysplastic or hypoplastic. They look a little bit smaller. They stay a little bit smaller, and they are a little bit smaller than wild, grossly cystic. And just to show you a comparison, here's the PKD1 knockouts using the same two Cre models, sized. They're not smaller, but you can see by P0, they're already big and they have lots of cysts in them. Ciliary signal. When we do the same thing with IFT88, you get the cilia-related outcome, not the centrosome-related outcome. Patient phenotype with regards to the small dysplastic, hypoplastic kidney phenotype in the CEP120 knockouts compared to the PKD1 knockout. And what's the effect on the progenitor of the cap mesenchyme? The 6B knockout is, of course, appreciating the two, so we see a reduction in the cap mesenchyme. And of course, in the PKD1 knockouts, either with 6-2 or HOXB, we don't see this loss of progenitor populations. And this loss of progenitors starts becoming evident around E17.5. At E13, no major loss. E15 also doesn't seem to be very pronounced. It's around E17. So it looks like it's slowly over time, developmental time, there's a loss of these cap mesenchyme cells in these knockouts. The next part of that, we wondered, well, as they're being lost, some of that is due to cell death, as I'll show you in a second. But is there specification happening? Is there fading happening correctly in these? So again, there are very well-defined stages of nephron development and multiple markers that can highlight renal vesicle stages and coma shape and S-shaped body stages of early nephron development. And so Tao went in with different markers, and he stained for these different structures so you can quantify the relative abundance per unit area. And he found something interesting. So again, at E13.5, no major difference. But at E17.5, in the two CEP120 knockouts, he sees more renal vesicles and more S-shaped bodies per unit area compared to the wild type. So it looks like these progenitor cells are prematurely fading or prematurely faded to become renal vesicles or S-shaped bodies. and developing. So they're undergoing this developmental paradigm quickly or too quickly. And it's this mix of cell death and mix of premature differentiation that we think is depleting the progenitor pool, which is resulting in the lower endowment or loss of nephron endowment in smaller kidneys as they develop. So then we said, okay, what about the other side of the coin? What about the ureteric bud epithelial progenitors? So what Tao did was he take the kidneys at different stages. This is E15.5, and he cleared the whole kidney and did light sheet microscopy on the whole thing so that we can go in and actually quantify accurately branch tips and branch nodes to try and get a quantitative measure of whether there's a branching morphogenesis defect in these kidneys. And you can tell these kidneys already look smaller. And when we quantify the branch tips and branch nodes, of course, there's a significant decrease in the abundance of both. So when you disrupt CEP120 in either progenitor population, you are losing the progenitor population, and that's causing nephron development defects and branching morphogenesis defects during early development. And then we said, well, what happens to these, what phenotypes do these kidneys have postnatally? And so between P0 and P15, now these kidneys start developing cysts and they become very fibrotic. You'll see that size-wise, they still aren't enlarged. They stay about the same size as a sort of normal wild-type kidney. And compared to PKD, which is already at P7, we see is very, very, very big and very, very cystic compared to these. And we can quantify the cyst index, and we see that the PKD kidneys have much higher cystogenesis than the CEP120 knockout. And both of these CEP120 mice die early. So we found that generally, they die around P17 or 18. That seems to be the most common day. They don't typically make it past P20, which is why we set P15 as our day to harvest the kidneys and analyze. Of course, we wanted the kidneys before the mice are dead. So we set ourselves a P15 time point, which is a few days before death. So within that window of time, now there's all of a sudden accelerated growth of epithelial cells to cause cystogenesis and accelerated growth of interstitial fibroblasts that are driving fibrosis. And here's the measure of fibrosis. We stain the kidney sections with anti-alpha SMA and several other factors. I'm showing you SMA here. The CEP120 kidneys have almost twice as much fibrosis as the PKD1 knockout kidneys. So again, it more closely resembles the human disease phenotypes for this gene than like an ADPKD mouse or a cilia loss mouse model. So this was just all some basic phenotyping, of course, and we wanted to get some unbiased views of what pathways are affected when we knock out CEP120 and disrupt centrosome biogenesis that are driving these phenotypes. And so we've come at this using multiple different ways. We've taken kidneys at different developmental stages, and we've done either bulk RNA-seq to get deep sequencing. We've also done single nuclear RNA-seq to try and identify the cell types associated with those changes. And we just finished getting a data set of whole tissue metabolomics. And we're going through and integrating all three of these data sets to try and come up with a consensus for some of the main pathways that we think are disrupted upon centrosome loss of function compared to cilia loss of function in ADPKD and also in the IFT88 cases. So I'll show you just a couple of examples of this data. So for the embryonic time point, one of the nice things was that the Cree models also expressed GFP, which gave us a way to then fact sort the centrosomeless cells away from centrosome-containing cells. So we could take HOXB Cree GFP only, or HOXB Cree CEP120, take the GFP positive cells, fact sort them away, then do bulk RNA-seq. And what that did was give us about 400 genes that are differentially affected, about 300 up, 100 down. We did pathway analysis, and you can see these over here, and I'll take you through some of the validations and what some of the pathways mean. So one of the first things that jumped out at us, and you can see that the most impacted component in this is the mitotic spindle. And you'll see P53 and G2M checkpoint. These are all related when you look actually at what pathways are implicated. And again, I'll point out that these things don't come out if you just look at ciliary signaling or ciliary structural protein defects. This is a centrosome-associated phenomenon, and this kind of defect you would only see in the mutations that disrupt centrosome function. So why did these three categories pop up? Well, we know that centrosome number is tightly controlled by many checkpoints in cells as they divide and grow. And so if you have abnormal centrosome number, and I'll just take you down this path right here, the centrosome loss. So our mutation, which causes centrosome loss in CEP120, that causes delayed mitosis and abnormal spindle formation. These delayed entry or passage through the metaphase-anaphase transition. There are these two effector proteins for p53 that end up stabilizing p53, which results in either cell arrest or cell death. This is called the mitotic surveillance pathway for centrosomes, okay? And so this, again, this is something that you wouldn't encounter if you were just thinking about the cilia-associated mutations. This is a very centrosome-centric pathway. And so the centrosomal mutations that drive these kidney phenotypes are going to have these defects, whereas the cilia ones will not, okay? So that's one of the take-home messages. We also went in and validated, of course, we can see by IF staining that p53 is accumulated in these centrosomal cells, and these cells are dying. They're cleaved caspase 3 positive. We can then do immunoblotting again to show this p53 activation, and also to validate some of the other pathways that come out of this. As you can see, the most downregulated is OXPHOS and metabolic processing. So there's a lot of mitochondrial defects. There's TNF alpha signaling. There's a lot of Wnt signaling defects that we think are driving those early fate determination decision-making tree defects that I showed you during early development. So we've been able to identify and validate several defects that we think are causing the congenital kidney developmental defects. And then we said, well, what about postnatally? What pathways are affected there? And for this, we took the whole kidney. In this case, the Cree's expression of GFP is no longer active. So we couldn't FACS sort and enrich for the centrosomal cell. So we had to basically just mash up the whole kidney. And so you gain a mix of centrosome containing and centrosomal cells. So the signal to noise is not as good. So we got a lot less DEGs, just under 100. And when you look at those, again, energy metabolism significantly affected. And the main pathway was EMT. Now, this is not classic EMT. Actually, when you dive into what's in this category, it's a lot of cytokine receptor inflammatory signaling types of pathways that are affected, okay? And so to quickly summarize this first part of the story, basically, what we found is that when we knock out CEP120 and induce centrosome loss in the developing kidney, we see changes in Wnt signaling in the different compartments than what we're supposed to be. We see decreased renewal of the progenitor cells, whether it be the mesenchymal cells or the UB cells. And this is partly due to increased p53-mediated mitotic surveillance pathway-associated cell death, which is depleting some of these progenitor cells. Then you have this premature differentiation that is, we think, is driven partly by aberrant Wnt signaling. And so it's the combination of cell death and premature fading that's depleting these progenitor populations, which is causing reduced number of nephrons and nephron endowment defects, smaller kidneys. But interestingly, these cells, once they're faded, can now proliferate and form cysts and cause fibrosis in the stroma, et cetera, et cetera. So there's this switch between how cells handle centrosome loss when they're in the progenitor phase versus when they're in the differentiated phase, which I'll touch upon in a little bit again. And again, in the PKD1 loss, using the same two Crees, we didn't see any of these particular defects. Kidneys develop normally, it's just they grow and they become cystic very quickly, and they have all the secretion defects, et cetera, et cetera. So what about the knockouts in the stroma? Everything I've showed you so far was in the nephron populations. So this project was driven by my other postdoc, Ava, who's now at UT Southwestern. And so she used the FOXD1 Cree. Again, this is just a developmental Cree, not inducible. And she crossed it to the CEP120 flox. And the idea was to look at what happens to the progenitor cells and what happens to the cells that are derived, the stromal populations of cells that derive from these, and also what's the effect on nephron development, because we know the stromal progenitors can influence nephron maturation and timing. So Ava crossed these mice. She could confirm SEP120 loss and centrosome loss in the stromal progenitors. And then I'm just going to summarize for you quickly some of the main take-home messages from this paper. These mice were also sickly and a little bit runted. A lot of the mice died early, so that you can see this mortality curve. There was a lot that died before P20. These are short-term survivors. We don't think these mice were dying because of kidney effects. We think these were not related to the kidney knockout effects because the kidneys look mostly normal. Okay, there's this medullary atrophy that we see here. There's some tubular dilations in the medulla region over here, but there's no cysts, there's no spontaneous fibrosis, and there's actually no decline in kidney function, which is why we're pretty sure that these mice are not dying because of the kidney defects. We know FOXD1 is expressed in other organs, including the brain, and it's very well known that disrupting centrosomes in the brain will cause early lethality in mice. So we think that's what's happening. But some of these mice make it past that. And these are the long-term survivors, and we can actually follow those and study them. Those also don't show decline in kidney function. So it tells me that knocking out SEP120 and centrosomes in the stroma, at least grossly, doesn't induce cystogenesis, nor fibrosis, nor does it disrupt kidney filtration function, which tells you which cell types we should maybe focus on for the NPHP-RC with mutations in these categories of genes. But of course, we wondered, well, what happens to the interstitial lineages of these cells when we ablate SEP120? So again, in the absence of SEP120, the progenitor cells will lose their centrosomes. What we found is that these cells, again, undergo that P53 mitotic surveillance-based apoptosis. And so you get loss of a good chunk of the stromal progenitor population. You get less differentiated interstitial cells. Even some of the mesangial cells in the glom are reduced. We also see delayed nephron tubule maturation, although nephron endowment in general looks okay. And they catch up, meaning it's just a delay in maturation. It's not a block. So when we leave these mice, those long-term survivors for a few months, everything looks normal with regards to nephron development. And there's this slight kidney reduction in size. So, but then we thought, okay, if there's no effect on cystogenesis and spontaneous fibrosis, what is there a contribution if the kidneys are injured and undergoing repair? Because one thing I didn't show you from the previous study was we also did injury experiments in those 6-2 and HOXB mice. And in those, in the SEP120 flox mice in the adult case, and we see rapid cystogenesis and fibrosis post-injury. And so we thought, does this also happen in these stromal knockouts? So what Ava did was she took these long-term survivors at P60, did UUO, and then harvested the kidneys 7 days later. And so up top here is the sham, down here is the UUO, here's the control, and here's the knockout. So you can see that the UUO, of course, induces an injury response when you look at desmin and fibronectin, but it's elevated when you knock out centrosome. So it looks like there is a hyper injury response. These cells are hyperproliferating and causing enhanced fibrosis when you ablate SEP120 from these kidneys. So to put the two stories together and sort of give you a summary of our general phenotyping so far and which of the population of progenitors with a mutation in this centrosome gene category have a problem in. So if you look at abundance of progenitor and derived cell types, if you knock them out in the nephron progenitor, the UB, or the stroma, you get reduction in those progenitor populations in both. For branching morphogenesis, we saw a defect when we knocked it out in the nephron or UB progenitors, but it was normal in the stroma. Kidney size and nephron endowment was very affected here, only slightly affected in the stroma. There's spontaneous fibrosis in the NPC and UB knockouts, but no spontaneous fibrosis in the stroma. There's definite cystogenesis. It's very, very mild in the stromal knockouts. Kidney function is definitely affected in the NPC and UB knockouts, but is normal in the stromal knockouts. and fibrosis and cystogenesis post-injury occurs when you damage them. So you start getting an idea of which cell types might be playing a role during development versus during injury and repair, and where we should sort of think about therapeutic targeting and which cell types might be more important than others. And finally, to put this a little bit into context, different organs handle centrosome loss differently. So for example, this, I told you earlier, this is a big problem in the brain where a lot of these mutations cause microcephaly. So these progenitor cells, if you have a mutation in the centrosome gene, they will activate this mitotic surveillance pathway, and these progeny will undergo apoptosis. You will lose the stem cells, and so you have less of them to form neurons, and that's why you get less neurons in a smaller brain. But that's usually the end of the story. Those neurons that have differentiated don't then proliferate to form tumors or do anything else. You're just stuck with that small brain phenotype. In the lung is a kind of a weird mix of the hybrid of the two things. It depends on where the mutation is. So if you cause a centrosome gene mutation in one population of progenitors in the lung, you won't have an effect. They will continue to grow just fine. But in another population, you will see an effect. In the kidney, it doesn't seem like the different progenitors behave differently. Loss of centrosomes in the progenitor cells, all three progenitor cells, blocks their overall growth, their proliferation, and their proper fate determination. Okay, so that's consistent. But the difference with the kidney and these other organs that have been studied is that once the cells differentiate into tubular epithelia or the stromal cells, for some reason, now this mutation in a centrosome gene conveys a proliferative advantage to those cells. Now those cells can actually hyperproliferate and form the cysts and form the stromal fibroblasts that are causing fibrosis, unlike these other organs. So there's something different about the switch in the kidney that we're trying to understand. And so we've been trying to think of ways to target these pathways now, both the early developmental defect and the postnatal fibrosis and cystogenesis defects. So one of the pathways we're trying to target is this mitotic surveillance pathway. You can't target P53 because you're going to mess everything up. But several groups have shown that this one protein, USP28, is very, very specific in regards to its function to this mitotic surveillance pathway associated with centrosome loss. And there's been a few studies now where people have used this as a handle to disrupt this pathway and rescue centrosome loss associated cell death. So we're planning on using genetic models like the mice and small molecule inhibitors in our CEP120 knockouts to see if we can rescue those defects. So the experimental setup is that we take our CEP120 loss and take models of ciliary signaling loss or ciliary structure loss and inhibit this mitotic surveillance pathway to see if we can rescue the kidney developmental defects. And then we also trying to do this in vitro. We got lucky. We got primary cells from those patients with CEP120 mutations. We were able to reprogram them into iPSCs and have grown kidney organoids from them. And so we can go in these organoids and disrupt this pathway again as a model of early nephron development and see if we can rescue this. On the flip side, two of the most upregulated pathways that came out of our postnatal fibrocystic kidney disease stage was this cytokine receptor axis, CXCL5, CXCR2. It's been heavily associated with inflammation and fibrotic diseases in lots of organs, including in the kidney with regards to AKI and CKD, but it hasn't been studied in the context of nephronophthisis or the NPHP-RC. But it's really one of the most highly upregulated signaling pathways in our knockouts. And MMP9 is a matrix metalloprotease, also implicated in cancer progression and fibrosis, also implicated in the kidney in AKI and CKD, but not in NPHP or NPHP-RC. We've mined the single cell omics data. They're expressed in nephron epithelia, in the stromal cell fibroblasts, in immune cells. So it can be in multiple different cell types. And we've come up with this model, which is testable, and we're working on testing it, which is that in the absence of centrosomes, different epithelial cell types will secrete these factors like CXCL5, which can function on stromal populations, immune cells, and in an autocrine way to drive cystogenesis of the tubular cells or fibrosis of the stromal cells, and of course the immune cells that exacerbate both of those phenotypes. And so we're doing testing of these pathways, again, using floxed mice and small molecule inhibitors of either CXCL5, CXCR2, and MMP9, and doing it in vivo in mice and in organoids. And just as a teaser, this is all very, very preliminary. But basically, we're using these small molecule inhibitors. We're seeing that we can rescue this cystic and fibrotic effect of the CEP120 loss when we treat those mice with these inhibitors. We see improvements in cystogenesis, improvements in fibrosis, and we can extend the life of these mice, at least so far, a little bit. Okay, I know I'm going a little bit long, but I really wanted, since this is the PKD Resource Center, I wanted to tell you a little bit about this new imaging modality that my lab's been working on. My lab uses a lot of microscopy, and over the years, we've developed applied a lot of different imaging methods to study centrosomes and cilia in the kidney and different tissues. And expansion microscopy is really a really cool new tool. I think a lot of people should give it a try. It sounds technically challenging and daunting at first, but actually I would say it's really worth trying. And we wrote a protocol to try and help people in the nephrology field get past this activation energy. So the concept is really simple. Unlike super resolution systems that super resolution microscopy tries to use fancier and fancier optics, really expensive systems, to try and resolve the localization of two proteins or two molecules that are close to each other. Expansion microscopy says, let's forget about the microscope, let's do something to the specimen. So in this case, you physically expand the specimen by embedding it in a hydrogel and then swelling the hydrogel so you can separate the two molecules in space from each other in 3D space. And then that helps you improve and be able to resolve where these things are. So this, 2015-16 was when this method was first established by the Boyden Lab. And a couple of years later, along with in a collaboration with Jadranka Lonkarik at the NIH, we published a story where we applied a different type of expansion microscopy for analysis of centrioles and cilia to show that we can basically expand these structures and do immunofluorescence imaging that gets you near electron microscopy level resolution without needing any really fancy microscopes. And for the aficionados who have worked on expansion microscopy, this particular one is the MAP, the magnified analysis of the proteome method. And in this paper, we'd used it on cells only. So cultured cells, RPE cells, IMCD3 cells, and we'd also done it on airway multiciliated cultured cells. So what we wanted to do was take this and apply it to kidney tissue. There's been a lot of labs now that have applied it to all kinds of organs, and even for the kidney, they've done it on whole kidneys and different methods. There are different protocols for expansion microscopy that use different chemistries, different approaches. We've tried a few different ones. We think this one works the best. This story was published recently. It basically gives you a step-by-step how-to cookbook to take a kidney, either from mice, but also clinical samples from humans, from patients, which was really important to us. Basically, if you can get a block of tissue, we can tell you how to go through and do the gelation steps, the denaturation steps, the expansion, punching, and staining. And there are many advantages, of course, to this protocol compared to traditional imaging. First thing, of course, is you don't need any fancy microscopes, especially if you're in a clinical setting and you have regular, small, low objective, low magnification objective lenses. You don't need any fancy microscopes to get really nice imaging. It works on both mouse and human kidney specimens, fixed in all kinds of different ways. So we tested different varieties to make sure that, depending on how you get them from the clinic, they could be useful. This method improves antigen accessibility. So actually more antibodies work than don't, because there's an antigen retrieval step in this. You get multiple staining tests from a single tissue. So if you have rare, 1 or 2 specimens from one rare patient, you can do multiple staining combinations from that one. And there's lots of other small advantages that we wrote in the paper so you can read about and see what you think. We wrote it like a book chapter. So it has all the tables with the product numbers and the concentrations and the catalogs and everything that you need in a step-by-step how-to. We try to simplify it as much as possible with all of the details so you don't have to go figure out anything out. We've tried to include some helpful pictures on some of the steps where we think there may be, a visual aid would help you get past that step. And then, of course, we had to go in and using internal structures like centrioles with a well-defined size as a molecular ruler, so we can measure and make sure that there's the expansion coefficient is working. So just as an example, here's a confocal image of a 60x objective stained for cilia and E-cadherin and nuclei. This is what it would look like. This is what it looks like when it's expanded at 40x, expanded at 60x. So the same specimen expanded at 60x, now you can start seeing the level of detail differences. You can then take this onto bigger and better microscopes with 100x or super res systems like ARIScan. You can start seeing, structure in these centrioles and cilia. We can quantify their diameter and length and show that the expansion is happening isotropically and it's consistent with EM size. Basically, you can get EM-like resolution. We wanted to show that this can be usable for lots of different kidney cell types. So the typical markers that many of you use to stain your kidneys or counter stain your kidneys. So proximal tubule markers, distal tubule markers, collecting duct markers, podocyte markers. Up top here are all the unexpanded examples at 20x. You could see what, here's how gloms look at 20x unexpanded. Here's a 20x expanded glom. You can start seeing structure. Again, we used low magnification objective lenses as examples here, because in case you're in a clinical setting and you don't have access to anything better than this, you can still do some really nice imaging and analysis. But of course, if you have high objective lenses, you can really knock this out of the park. Many, many of these markers work. We listed them in the paper as well as their concentrations to try. For us, we're cilia centrosome nerds, so we wanted to know how many of those things can we stain for. We use different markers against different substructures of the centriole to see that we can indeed spatially resolve them to their unique compartment. This is all in tissue, I should add, right? So this is normally very, very difficult, if not impossible, to pull off even with super resolution imaging. If you really want to push this, you can take this to super res and combine it with expansion, and now you can really even see foot processes, and you don't need to do EM anymore to look at foot process effacement. You can use expansion microscopy with regular microscopes and analyze foot processes and see if your knockout has foot process effacement. And then, of course, you can apply it as a test. We went we expanded ADPKD kidneys. We can see the morphological defects in the cells that people have described become more pronounced. The cilia are longer, a phenotype people have described. We can quantify that nicely. Centriole abnormalities, which we published a few times, other people have published. The hyperacetylation of the cystic cytoplasmic microtubules, the ECM deposition of the stromal populations. So a lot of these things that you've been looking at and staining for, you can do at much better resolution using simple microscopes if you follow this protocol. We would love you guys to try it. Again, there's many, many papers on expansion microscopy. We wrote this for the kidney specifically. And if you run into any steps that are problematic or if you hit a snag, please write us. We're happy to work through it with you and hope that it works out for anybody who's trying it out. Okay, I'll stop there. Thank you all for your attention. The people who did the work that I showed you were Tao and Ava, who did the knockouts, and Ava also did the expansion microscopy. We have a lot of help in the nephrology division and our wonderful WashU ciliopathy group, our funding from NIDDK for a lot of this. Thank you all for your attention, and I'm happy to take questions.
Mo, wonderful talk. Thank you very much. I'm going to open this up for questions. So what I'm going to ask to foster communication is that you raise your hand and then communicate directly with Mo. I'm going to start out. Sorry, Jeremy, I just jumped right in front of you there. I'm going to start out asking a question. Sorry, I had to step away for 2 seconds, and I may have missed it during that time. So when you take away these centrals, I would assume you also have a basal body defect, which means then you're going to also have a ciliary defect.
Correct.
Um, how do you get the different results with those two things happening?
So that's exactly why we did the three experiments, the three models side by side. There's no way to get rid of a basal body but keep a cilium,
Yeah.
right? So the only real way we can do it is by deduction. So either you have your centrosome and cilium intact, but you're just missing polycystin. So that's the ciliary signaling defect. Or you have a IFT88, so the cilium is lost. You have your basal bodies. And then you have, of course, your basal body knockout, which also has lost its cilia. But the cilia loss model comparison to that will help us tell which ones are the centrosome-related defects. Does that make sense?
Yeah, it does. I would have expected that the basal body loss would be the most severe one because it's taken everything out, right?
It is the most severe one, I would
Well,
say.
I guess with cystic disease, I guess is what I'm referring to. But yeah, developmentally, yes, you're right. Thank you, man. Jeremy.
Yeah, super nice talk, super fascinating research as always, Mo. Like the expansion microscopy it looks incredibly tantalizing. But I had a question about the first part. I think that it's very interesting that P53 gets activated upon loss of centrosomes. Maybe not super surprising, and that maybe that's part of the etiology of some of the phenotypes that you're seeing. I understand that it's difficult to get rid of P53 entirely and have a normal looking kidney. So the epistasis might be difficult to work out like that. I was wondering if you've thought about like mildly misactivating P53 by like removing MDM2 or MDM4 and seeing whether that was sufficient to cause some of these phenotypes in the kidney.
That's a great point. We haven't done that experiment, but we're knocking out USP28.
Yeah.
And that you're essentially we're rescuing with that We're basically bypassing that mitotic surveillance activation of P53. And that was who recommended? I want to say either Andrew Holland or Hisham Bazi recommended that manipulation as being a little bit more surgical than P53, which is why we're doing that.
Got it. Great. Awesome. Makes sense.
Yeah. Thanks,
Hey.
Jeremy.
Hey, great talk. Thank you. I had some questions about the progenitor cell work that you did in the first part.
Yes.
When you remove the CEP120, what consequences do you see when you look at the progenitor cells on things like organelle distribution and ploidy?
Ooh, yeah. The ploidy we haven't looked at. Ploidy we definitely haven't looked at, but that's one that we absolutely think there's going to be a big defect in because of the mitotic segregation errors and the mitotic spindle assembly errors. We know these cause. So when you have centrosome defects, the spindle will still form as a survival mechanism for the cell. In the absence of centrosomes, actually cells will upregulate at all the mitotic spindle proteins. And if you notice in our RNA-seq, the highest upregulated pathway was mitotic spindle. It's a survival mechanism, coping mechanism for cells to form a spindle. But when they form a spindle, they make abnormal microtubules that you'll get all kinds of weird myriatylic attachments to the kinetochore. So you do get segregation errors. So we fully predict there's going to be some ploidy defects, but we haven't done the assay to look at what's happening. We just kind of expect it to be there.
Just a very simplistic question. When you imagine that unequal distribution that you predict, is it strictly the cells that lack the centrosome, that don't inherit it, that die of the activation of P53?
Yes.
It's strictly the ones that don't.
Correct. Only those ones internally activate P53, that surveillance pathway.
Yeah.
Now, of course, when you eliminate those cells, you're going to have effects on other cell types that depend on those, which is why, as I mentioned earlier, when we're activating P53 and knocking and sort of killing the mesenchymal cap mesenchyme cells, it was of course having an effect on the UB cells. So we were seeing some defects with growth and branching on the UB side, but that's because of the reciprocal signaling and not the direct effect of the gene knockout.
Yeah.
And I think part of the reason we did the experiments the way we did with teasing which, knocking it out in progenitors separately was to try and answer these kind of questions. Because if we just knocked it out in every cell all at once, it would be really impossible to kind of tease these things apart.
Sure. Very cute. Very, very cool study. Thank you.
Thanks.
Michelle.
Hey, Mo,
Hey,
beautiful.
Michelle.
I got two quick questions. The first one is the centrosome phenotype appears to be more severe than the cilia phenotype, but that is because of the developmental defects. So the question is, inducibly, once the development is finished, have you done those experiments, and does that now mirror the cilia defect?
Yeah, great question. So in parallel, when we're doing those experiments, Tau crossed the CEP120 flox to Ben Humphreys's proximal tubule SLC34A1 Cre-ERT. So that gives us control over when we knock it out, and only in the proximal tubule, right? So he knocked it out, and it's very similar to the old IFT88 experiments, for example, where you get really slow cystogenesis. We took 12 months, 14 months before we started seeing really prominent cysts and stuff like that. So it happens really, really slowly. So a lot of the defects are caused developmentally when you have this mutation.
Right.
But that's why we did the injury in those. So we took those SLC34A1 Cre, we knocked out CEP120, waited a month, there was no cysts or anything. And then when he injured those, now you get rapid fibrosis, rapid cystogenesis, less cysts than PKD with the same experiment, more fibrosis. So it's kind of, again, mimicking the disease, the patient phenotypes a little more closely than the cilia knockouts or cilia disruption.
That's great. Just a quick suggestion for that, perhaps the PAX8ERT2, which is what everybody's been using for the ADPKD cilia comparisons. If you did that with your centrosome deletions, that would give you a clear, apples to apples.
It would put a good in comparison to the rest of the work.
Yeah, exactly. But the second question I had was, as you are very well aware about the paradoxical effect of cilia deletion by itself versus cilia deletion in the setting of PKD1 deletion, Steve Samlo's work. And then, to layer that off with Massimo Attanasio and now Steve's work about GLIS2, removing that in the setting of ADPKD and that getting better too. And as you know, GLIS2 is an NPHP gene. It sits in the nucleus. Steve has done the cilia work. So the question is, can you implicate the centrosome, cilia centrosome nuclei using your CEP120 and show that improves ADPKD? You see what I'm saying? The epistasis between PKD1, CEP120 deletion. Have you thought of that? Are you thinking about doing it?
100%. And when Steve came here, he told me to do that experiment too. The reason we didn't do it is because of the fact that, we should do it. I should say, my prediction is 100% it would give you the same phenotype as the loss of cilia. Basically, I would predict we're going to get the Samlo result from that knockout, because in our knockouts, we're also losing cilia. So unless you think that losing cilia, but losing centrosomes now will somehow overcome that epistasis, I didn't think that would happen. So we didn't prioritize that experiment per se. But I think it's a valuable one to do ultimately, just to put it in the same category and say, yes, this also behaves that way.
Yeah, great. Thank you.
Last question, John. You're muted, John.
Yeah, okay. So, and Brad's going to hit me two questions. One is, I don't get the problem with crossing the P53. There's no phenotype in the P53 knockout until like 6 months when they start getting cancer. There's no kidney phenotype. There's nothing wrong with them.
No, there is. There are a couple of papers that knocked out P53 developmentally, and they show developmental kidney problems.
Really?
Yeah.
We have the animals in our facility, and they run around fine.
It was one of the first experiments we thought about doing, and I went in looking for kidney-specific P53 knockouts.
Because this is very, Cancer Anderson had the CEPJ mutant for the cilia neural tube defect, and they could rescue a lot of the phenotypes in a P53 null background in that case. Not all of them, but some of them. Anyways, the second question is, you did your RNA-seq analysis. It looked like you took a separation approach as opposed to where's the overlap approach. It might seem like, the pathways that were in common between these three different models might be the more likely the cystogenic type pathways, right?
Yeah, so what I didn't get a chance to show you was the comparisons with the other two models. So I just showed you CEP120 comparison to wild type. But we've done PKD at the same time, and we get a whole different set of things in PKD compared to centrosome. Some things overlap, but there's a lot of unique things between the two. And you're right, like which ones do we want to focus on more? The things that are common and are going to be kind of not unique to one disease, but can be targeted in both cases? Or do we want to focus on the things that are maybe unique to this category of renal ciliopathy and let the PKD people focus on the PKD related stuff? We've been trying to focus on the centrosome associated pathways to see if we can fix that problem.
All right, with that, we're going to have to bring it to a close. Mo, thank you very much for a wonderful, stimulating presentation. And I want to thank the audience for rigorous discussion. I think that was a lot of fun. So Mo, thank you very much, and see everybody back here at the beginning of January for the next one.
Thank you for the invite. Take care, everyone.